Oh, good evening, everybody, or good afternoon, good morning, wherever you are. Welcome to this OpenFlow cytometry session. There's a few people still joining, so we'll start fairly slowly. As you can see from the splash screen there, as usual, there are a couple of ways of getting in touch with us. You can use the chat function if there's any issues with sound, but if you've got any questions, best place to put those is into the Q&A section, and as they come through, we will try and answer them either as we're going along, or if we don't manage that, we will come to them at the end. So today we are going to be looking at multicolor compensation. And this is something we did in one of our previous sessions, but we did that using the Beckman Dickinson machines and FACS-DIVA. Today we're going to be using the Beckman Coulter CytoFLEX LX, which we introduced you to on our last session. But let us introduce ourselves, I suppose. My name is Derek Davis, and I am the Science Technology Platform Training Lead here at the Francis Crick Institute in London. And joining me, as usual, on these courses or webinars are, let me hand you to Rui.
Hi, everyone. My name is Rui Gardner. I'm the director of the flow cytometry core facility at Memorial Sloan Kettering Cancer Center here in New York. And I'll pass it on to Kathy.
Okay, thank you, Rui. I'm Kathy Daniels. I'm an ISAC SRL emerging leader here of my own accord to help out with education, and really happy to be here today to go over multicolor comp on the CytoFLEX. Thanks, Derek. Thanks, Rui.
Okay, so as usual, these classes, what we'll do is we'll start off with a little bit of the background, the theory, so that everybody's in the same sort of place. But we'll try to spend most of the time here actually doing some practical work on the CytoFLEX. So I'm going to go over the principles of spillover and compensation, and then I'm going to hand over to Kathy, who's going to use the CytExpert software on the CytoFLEX to first of all manually compensate some samples, and then look at automatic compensation. And we have a 5-color panel, which we'll come to a little bit later on. We'll tell you what we're doing. Now, as usual, whenever you come to any experiments, you've got your two main tools, and you've got your fluorochromes, your fluorescent reagents, and you've got your cytometer. And it's almost always the cytometer that you need to think about first, because not everybody has the luxury of access to a large core facility, which might have lots of different machines of different capabilities. Many times people will have just the one machine. You might be a small biotech startup, or you might be working in a clinical environment even, where there's just that one cytometer. So you need to know several things about that cytometer. You need to know the lasers that are in there, how many there are, and the wavelengths of those lasers, because that influences the fluorochromes that you can use. But it's not just the number of lasers, it's the number of detection channels that you have in each of those lasers, which will influence the number of targets, the number of analytes that you can measure. And as well as that, you need to know something about the optical filters that are in that cytometer, because this is going to help us, one, make sure that we've got the appropriate filter for the appropriate fluorochrome, but also give us an idea of maybe how much overlap there might be. And we know, don't we, that flow cytometry has changed quite rapidly, I suppose, in the last 15 or 20 years. We've seen the introduction of many different types of laser. We cover now the whole range from deep UV 325 up to infrared or near infrared lasers. We have many more fluorescent probes than we had 20 years ago, many more fluorescent proteins, which means that we can do very clever things like 16, 17 or more analytes in an individual experiment. And of course, sometimes that is very useful if you've got a heterogeneous population. As I've probably said before, I'm quite a big fan of doing single color experiments, but obviously you don't get quite as much information in a heterogeneous population. But when we start adding more and more fluorochromes, this is when we have to start thinking about the overlap we're going to get between them. Because I'm sure you're all familiar with this, the spectral viewers that we get online from many of the manufacturers of machines and reagents. This is fluorescein, and this shows us in the dotted line the excitation spectrum, and in the filled line the emission spectrum. The important word there, of course, is spectrum. And we know from looking at this that if we want to optimally excite fluorescein, we're going to use a blue or 488 nanometer laser. And remember, in most of our conventional flow cytometers, we don't measure the whole of the emitted light spectrum. We just measure a part of it, and that's where that optical filter comes in. So normally something like a 530/30 bandpass filter. So we're measuring 30 nanometers of width of that emission centered at 530 nanometers. And that's great if I'm just doing a single color FITC experiment. If I'm doing a single color PE experiment, I know from looking at my spectral viewer, I can excite PE very well at 488 nanometers, and its emission is shifted more towards the yellowy orange part of the spectrum. And I'll have another optical filter, something like a 580/30, that will capture the maximum emission of PE. And if I'm doing a single color PE experiment, again, that's not an issue. The issue comes when we start to combine those. If I want to use both of those fluorochromes together, and if I put the two single emission spectra there, but the two filters that we're using, you can see that the problem we're going to get is some of those fluorescent photons will end up in the PE detection channel. We know that some of those photons from FITC is going to emit photons from the excitation wavelength, way up into the infrared. Some of those are going to be in the sort of orange region of the spectrum. Now, the cytometer doesn't know where photons have come from. It's merely steering them through the machine with its optics. So we know that even in the absence of PE, I'm going to get some photons that are going to end up in the PE detector from my FITC. Similarly, I might get a small number of PE photons ending up in the FITC channel. And if I magnify that, maybe that looks a little bit more obvious. And this is the problem we have with fluorescence spillover. And it's something that we have to deal with in any multicolor flow cytometry experiment. And how does that manifest itself? Well, if we do a very simple experiment here, where we've just got three populations of beads in this case, a negative population, a PE positive, and an FITC positive. Now, in the absence of any compensation, if I were to look at the histogram of the the FITC channel, I would see something that looked like that. That's great. I see my FITC positives, both my negative and my PE population are negative for FITC. But if I were to look at a histogram of the FITC channel, it would look something like that. I'd see three populations. Now, I should only see two, shouldn't I? I should see the PE positives and the things that are FITC positive and the things that are negative, because neither of those has any PE on it. So what we do is we perform fluorescence compensation, which is a mathematical transformation of the data, which you see on the right-hand side. And if I, again, draw my FITC histogram, I get what I expect. But now, at the end of compensation, I draw my PE histogram. Not very well, but I have drawn a PE histogram. Now I see the two populations that I want to see. Clear my drawings. And as we discussed in a previous webinar, so I'm not going to go into this into great detail here, compensation is generally expressed as a percentage. What we do is we look at the signal that we're getting in our FITC channel for each cell or each event that goes through the cytometer, and we look at the signal for that same event in the PE channel, and we work out what that ratio is. That then allows us to calculate a percentage, which we can apply in our compensation matrix. And what happens is that we bring our positive population, our FITC positive population, here in line with the negative population. And this is the end point of compensation, that both that negative population and that FITC positive population will have the same fluorescence intensity in the PE channel, the spillover channel, because neither of them have any PE in them. So how do we calculate compensation? Well, in effect, we're going to in effect, we're going to calculate the slope of this line. We're going to look at the median fluorescence intensity of the negative population, the median fluorescence intensity of that positive population, and we're going to adjust compensation until those medians are aligned. That's when we know we've correctly compensated. And we generally use the median of our population rather than the mean, because the median, as we've discussed previously, is a little bit of a more robust statistic than the mean. If I've got the odd outlier, that will affect the mean much more than it does the median. But you can use either. So how do we know when compensation is correct? Well, one thing we should never be doing is setting compensation by eye. Now, as we often say, we're very visual creatures, and flow cytometry is great because it gives us visualization of the data. So if I go back one slide, it sort of looks like these two populations are aligned. But we don't just want to rely on that visualization. We also want to get some mathematical proof, if you like, behind that to show that compensation is correct. And we'll see this in action in a moment. But what we're going to do is, in this particular example, I've just got a negative and an FITC positive population. So obviously I want to compensate the FITC signal from the PE channel. So in my software, we're just going to put some sort of region. It doesn't really matter what shape you use, but we're going to isolate the negative and the positive population. And here we've got no compensation set. And what we're going to look at is these two figures, the median fluorescence intensity of the negative, which is about 90, and the median of the positive, which is about 1900. And then in our software, what we're going to do is we're going to adjust this figure until we get to this point. So at 30.4% compensation, the median of the negatives and the median of the positives now have the same value. And that's what we expect. That's the end point of compensation. Now, obviously, to set compensation, you have to come to the cytometer with the right controls. So there are three rules of compensation. I always put rules in inverted commas because not everything is completely unbreakable, but three rules. And as long as we get those rules right, our compensation life will be as perfect as we could get it. One thing to think about from the outset is with compensation, we're just looking at the fluorescence. We're looking at the spectrum of the fluorochromes that we're using. But obviously, to run them through a flow cytometer, we need to attach those fluorochromes to something. So we can use either cells or we can use compensation beads. Many of you will be familiar with these. They're antibody capture beads. If I can, I will generally use cells to set my compensation, but beads certainly do have their place, and that's what we're going to use today, in fact. And we'll come back to those in a second. I said there are three rules of compensation. So what are those rules? And what I would say is the first rule, the primary rule, is that with compensation, we have to use the same fluorochrome that we're using in our sample. So we can't, for example, substitute Alexa 488 in our control if we're using FITC in our samples. Although they're both green, that's not really what we're going to be looking at in flow cytometry. We're looking at where those photons end up in other channels, because we're looking at the ratio of the photons that fall in the PE channel to the ones that fall in the FITC or the Alexa 488 channel. So we have to use the same fluorochrome. And also we have to treat our samples in the same way. Again, that seems obvious, but particularly if you're using beads, if your samples have been fixed and permeabilized, your beads also have to be fixed and permeabilized. I get into the habit of running my controls through the whole protocol. It's the same protocol as my samples, so they've all been through the same steps. In that way, we're controlling for any variation that we introduce by the way we process our cells. Second rule of compensation is that our positive cells in our compensation tube should be at least as bright as anything that we're going to get in our samples. And that's because we're trying to get those two, the positive and the negative populations, as far apart as possible. The further they are apart, the more robust the mathematical calculation of the slope of that line, which is what's going to be the percentage. And also we should acquire sufficient events. And in practice, that means probably make sure you acquire at least 500 of your positive and your negative events. Ideally a few more than that, but 500 should be enough to give you a robust calculation. And then the final rule is that before you stained your cells or your beads, the positive and the negative population should have had the same level of autofluorescence, because that's what we're going to use as our background in the spillover channel. So I said you can use cells or beads, and always where you have that choice, you have to weigh up what the pros and cons are. So if you're going to use your cells, one thing that you need to think about is, is the marker that I'm looking for as bright as possible? Because, I need to get those two things as far apart as possible. And also, are there sufficient positive cells? If I need to acquire 500 and I'm looking for a very rare population, it might take me a while to do that. And also another important consideration is, can I actually afford to use my cells as controls? Sometimes you may be sample limited. You don't want to use your cells, in which case this is where we're going to use beads. But there are still some questions there. Beads are very easy to use, but often they are restricted to certain isotypes. Most of the time we use IgG antibodies in flow and immunostaining, not always. Some of these capture beads are species specific. Now, most people are dealing with human or mouse or rat or hamster. These work perfectly well, but I've given courses at veterinary institutes where it's much more difficult if you've got other species, sheep, cattle, and so on. And of course, because they're antibody capture beads, they're useful if you're using antibodies in your panel, not so useful if you're using fluorescent proteins or other fluorescent probes. However, today, this is what we're going to be using. We've teamed up with Slingshot Bio today, who provided us with these SpectraComp beads, and Kathy will talk a little bit more about those when we come to the practical part in a moment. Another question, I would like to put this slide in again. I know we have discussed it before, but it's important to run your compensation controls every time, and not just run them every time, but prepare them fresh every time as well. Because otherwise you're not controlling for some variables. You might have a variation in your cytometer. Somebody may have changed a filter without telling you. The engineer may have been in. There may have been a new laser put into the cytometer. If you're doing a longitudinal study, your autofluorescence may change over time. Probably the most common reason that we should do it every time is because you've introduced a new tandem batch. And as we know, tandem fluorochromes are very useful to us, but we have to be a little bit more careful with them. Even the same antibody from the same manufacturer, if it's a different lot, may have slightly different spectral characteristics, and that would affect our compensation. So what are we going to be using today? So we introduced the CytoFLEX LX during our last session, which seems like a long time ago now, possibly was. Looks a little bit like this. And remember, one of the differences between the CytoFLEX LX and many of the other cytometers on the market is it doesn't use photomultiplier tubes to capture and measure the light signals. It uses avalanche photodiodes. Now, these work in much the same way. They amplify the signal that comes from the photons that hit the photocathode of the APD, but they don't have the same voltage change that we can do in photomultiplier based machines. And in the CytoFLEX, we use a gain to amplify the signal, which is a mathematical transformation again of the signal, and the voltage across the anode and cathode of the APD has been optimized by the manufacturer. But the way it collects light will be very familiar to anybody who uses a flow cytometer. We're using a whole series of filters, long pass filters, short pass filters, mirrors, and band pass filters to select the wavelengths of light that we're interested in. So as always, when we're designing an experiment, I like to lay out the cytometer something like this. So the particular CytoFLEX we're using, which is at the MSK Flow Lab, is 21 color, has 6 lasers, as you can see, ranging from a UV to a near infrared laser. And so there are 21 different parameters that we could measure there. And you can see that that's 3 detectors from the ultraviolet laser, 5 from the violet, 3 from the blue, 5 from the yellow, 3 from the red, and 2 from that infrared laser. So this is going to help us as well when we start thinking about spillover. Now, spillover is the thing that we're going to assess and account for with compensation. And there are 3 main sources of spillover. That is when we're using fluorochromes that are using adjacent channels that are very close together in the electromagnetic spectrum. The tandem dyes, of course, give us not necessarily problems, but things we need to think about. We're always going to see some residual fluorescence from the donor dye in a tandem fluorochrome. And because in our tandem fluorochromes, we are using 2 fluorochromes together, often the acceptor part of the tandem dye is going to be excited by a different laser. It's a fluorochrome in its own right. So let me just show you a couple of examples of where we might expect to see overlap. Adjacent channels. Now, if I've got a panel where I have PE, PE CF594 or PE Dazzle or any of those equivalent fluorochromes, I know that they're going to give me a good deal of overlap. PE emits at about 580. This one obviously emits at 594. So although I can use them together, I know that I'm going to have to compensate the spillover. Tandem dyes, PE size 7. With the tandem dye, remember, we're using the yellow laser in this case to excite PE, which transfers its energy to size 7. And size 7 emits at about 780 nanometers. But that transfer of energy is never 100% perfect. So we always see some residual fluorescence of the donor in the tandem dye in its channel. So we would see some fluorescence in the PE channel. So if I've got that combination of fluorochromes, I know that I may have to address the spillover that I get. And then example of the acceptor dye, PE size 7. Of course, PE size 7 is preferentially going to be excited by the yellow laser. But as that cell with the PE size 7 passes through the red laser is not going to excite the PE, but it's capable of directly exciting the size 7 molecule. And of course, the size 7 molecule is still going to emit at about 780, which is where we would expect to see APC size 7 fluorescence. All of these cases of spillover, we're going to be able to deal with them by the process of compensation. It doesn't matter where that spillover has come from or how it's arisen. We use exactly the same process to account for it. So let's think about the live run that we're going to do today. And again, thank you to Slingshot Bio. Actually, I should mention that at some point in the reasonably near future, I'm partnering with them to run a webinar, and they produce these sort of artificial cells. So we're using the TruCytes TBNK kit, which, as many of you immunologists will know, that that's looking at T cells, B cells, and NK cells. So we've got a 5-color panel here today, looking at CD45, which is going to pick out all the mononuclear cells, CD3, our T cells, CD4 and CD8 subsets of T cells, and then CD19 are our B cells. And as you can see, the fluorochromes we're using there fits the Cytex Fluor 420, PE, Brilliant Violet 605, and APC. And we've employed the services of Anna and Chelsea at the MSK Flow Lab. This is a bit of a departure for us today because I'm in London, Kathy is somewhere near Boston, and Anna and Chelsea and Rui are in New York. So just before I hand over to Kathy, we'll just quickly look at where those fluorochromes are going to be detected on our CytoFLEX. And you can see we've sort of spread them out, but we have 2 fluorochromes from the violet laser, our CD3 and our CD8. This is BV605, and hopefully many of you are aware that most of the BV dyes, certainly above 520 nanometers, are tandem dyes. So BV605 the donor part of that is BV, brilliant violet 421, which is going to be emitting here. So by looking at this layout, we might see where we potentially could have some overlap problems. We haven't made it too complex an experiment here today. So that's the preamble. Let me hand you over to Kathy. I'll stop my share.
Okay.
And just a reminder, as Rui has put in the Q&A on the chat there, yeah, please feel free to ask as many questions as you can as we go along. This is your chance while we're here live to do that. Over to you, Kathy. Sorry.
Okay. No, perfect. Thank you so much, Derek. And I think we're ready on our end for the CytoFLEX screen to go ahead and start sharing. So I just want to make sure I really reiterate Derek's point to say thank you so much to Anna and Chelsea at the MSKCC Flow Core Lab. So they did all the hard work. I'm doing the easy work by doing a remote demonstration of how to run compensation and a multicolor experiment on the CytoFLEX LX using CytExpert. But they did all that heavy lifting, like I said, and we wouldn't be able to do this session today without them. So here we are. We're in CytExpert software. And what's going to be a little bit different about running a multicolor experiment using the CytoFLEX and using CytExpert is the approach that we take. On a lot of instruments that we're used to, if you haven't worked with the CytoFLEX before, the single color control acquisition and running compensation is typically done within the same experiment as your experimental samples, okay? When we're running on the CytoFLEX, we actually go ahead and carry out a whole separate compensation experiment that will then be imported into your actual experiment. So in order for us to get started today, to demonstrate the multicolor run here on the CytoFLEX, what we're first going to have to do is we're going to have to do a new compensation. So if you can see my mouse, I'm going ahead from this middle window, and I'm going to generate a new compensation, and I'll call it compensation OpenFlow 100722. And then from there, I'm just going to go ahead and save. You could, of course, change your file path to wherever you'd like it, the standard that you would expect on any instrument to be able to direct it wherever you need to go. All right. Now, what I typically like to do is a little bit different than what some other people might like to do. What we've spoken about in some prior sessions when we've talked about voltage and gain optimization is making sure that your fully stained experimental sample is on scale and that you're getting the best possible resolution of your populations. So we want to do the same thing here utilizing the CytoFLEX, but it can be slightly
Yep.
I think we may have lost Kathy there. That was the first time we've had a glitch like that.
I guess we should.
We should yes, sing and dance, Rui.
Yeah, we did that.
Any questions that people
but actually
have?
there's a question that we can.
So it looks like I got kicked out there for a second when I changed my view. I'm sorry, was everyone else still here? And then it was just me?
Yeah,
Yeah,
I
no,
think it
no.
was just
Okay, that was great.
But there is a question here.
Okay, perfect.
From Nick Chen. With CytoFLEX, do you still suggest preparing comp samples every time we run if we use the same lots of staining dyes?
That's a phenomenal question, and that allows me to ease back into my presentation, so thank you. So running a single color control each time that you plan to have an experiment is critical, right? So that's a conversation that we often have in the lab, and it's something that I'm very firm in my beliefs about, because you have to take into account that even if the instrument is performing the same exact way from day to day, you can have differences in reagent performance, right? So you want to make sure that you're running your single color controls at every run. I can't tell you the amount of times that people have come to me and the compensation has been off because they've used what I use terminology for is recycled compensation, and things are off and there's no way to recover it, because I say, okay, I can likely identify what the issues were here. Let's see your single colors. And they say, oh, I don't have those. I ran it back 2 months ago when I first did this experiment. Okay, so we definitely want to run them each time. Right. Now, what I'm not going to do is I'm not going to change my zoom ratio, because that seemed to have caused everything to quit before. So let's go back into the trick that I mentioned before, right? So I mentioned that in order to optimize our gains for this multicolor run, what I needed to do was I needed to run a little bit of my fully stained experimental sample. So in this window that we see here, you have your unstained cells, unstained beads, you have every single detector, and you have the capability to check off which controls that you have in a compensation experiment. There is no option to look at a fully stained experimental sample. So what we're going to do is we're kind of going to cheat the system a little bit. So what I know is I know that in well B2, we have our fully stained TBNK TruCyte controls from Slingshot. So what I want to do is I want to run those first and make sure that I have the appropriate gain set up for this experiment. So what I'm going to do is I'm going to go ahead and select one of the detectors that I'm using for this experiment. So we'll say that's blue 525. So the way that this instrument is set up is the detector is named after the excitation source. So blue, a 488 nanometer laser, and 525 is the midpoint of emission for the collection filter. Okay, so that's going to be for FITSI. But what I'm going to do is I'm going to say, all right, this is going to be in well B2, and I'm going to set that as my sample well. By doing that and setting that as my sample well, what I'm doing is I'm tricking the CytoFLEX into thinking that that's where I have my FITSI single color control, even though it's really my fully stained experimental sample. Then what I need to do is I need to change my plate type to make sure it's the appropriate plate that we're using for this experiment. So the plate type on this experiment is going to be a 96 well U bottom. Okay, and it just gives me a warning that I want to make sure that the choice of plate type that I've used is appropriate. So I'm going to say, yes, I do want to go ahead and make that switch. I wasn't using a flat bottom. I do want to use a U bottom. And that's something that we typically use in flow cytometry acquisition experiments, because you typically tend to have a lower dead volume with U or V bottom plates than you do compared to the flat bottom plates. Okay, so we're there right now. We have our one well selected where we have our fully stained sample, and I'm going to go ahead and I'm going to select OK. What you'll notice is that even though I only selected that one detector, what it's doing is it's generating a forward and side scatter dot plot in addition to a histogram for each and every single detector that's on the CytoFLEX LX, even if we're not using it for our experiment. Okay, so what I know about my experiment is, and what Derek had shown with his slide, is that we're going to be using FITSI. So I know I need to be on the lookout for the blue 525 detector and making sure the signal is appropriate there. I also have CD4PE, and that's our yellow-green 585 here. So go ahead, and I'm going to make that a little bit bigger. Can make this a little bit bigger as well. I'm kind of just going to shadow the rest of them. Then I have CD3 C-Fluor 420. So that's going to be our violet 450 right here. And I looked all this up in advance, so I know that these are the excitation and emission ranges that I'm expecting. So they're matching up with the detector. Okay. So I'll just leave that off to the side as opposed to making it bigger. Then we have our CD8 BV605. Okay. So I made that a little bit bigger as well. And then we have our CD19 APC, and that's going to be this red 660. Okay. So these are the detectors that I'm going to be on the lookout for making sure that we have the best possible separation. Okay. Another way you could approach this, if you had wanted, would be to go ahead and move all of the other histograms down lower so that you're only really seeing the ones that you want to see up top, and the ones that you don't want to see, you could have them a little bit lower. Just being a little bit stubborn, I wanted to make sure the ones that I wanted to see were a little bit bigger. So I got that violet 450 to get slightly larger on screen. Okay. So then what I want to do from there is make sure that I'm visualizing enough events when I go ahead and I start my acquisition. So under events to display on the left side of the screen, I'm going to change that to about 10,000. Or if I wanted, I could go a little bit higher. I can even go to 20,000, right? We want to make sure that we're seeing enough events that I can accurately determine if all of my events are on scale and giving me a decent resolution. And then I want to check my sample flow rate. So what you'll see on the underneath the events to display and all of our recording settings is you have your sample flow rate, and you can go from slow to medium to fast, which is about 10, 30, and 60 microliters per minute. Something to bring up that might be very helpful to people is to understand that you can get volumetric measurements as a readout on the CytoFLEX, but what you have to do is you have to make sure that you've calibrated the flow rate in advance of doing an experiment where you need to have an understanding of what the volume was if you're trying to do counts. And it involves measuring the weight of a plate that has wells filled with water, both before and after acquisition, so it could make sure that the volume uptake makes sense in correlation with the sample flow rate based on the density of water. Okay. So then from there, I'm going to go ahead and I'm going to go to initialize. And for those of us who were able to join us, or those of you who were able to join us for our last session, what you'll remember is that we have different methods to be able to go ahead and optimize our gains for the best resolution on the CytoFLEX. One option is to open this acquisition settings window right here, and you're going to see all the gains for each and every detector. So for me, and I see Rui there, so I'll stop in just a second. For me, seeing all of those different gains can be a little bit overwhelming, right? You can see that the range is between 1 and 3,000. And I believe on this instrument, there's about 21 fluorescent detectors, and then you have 2 scatter. So that's quite a bit to look at, even if I only have 5 fluorescent detectors that I need to worry about for this experiment. The other option is using what I like to call the G hand or the gain hand in the software to modify some of those settings. And that's what we'll do today. And I will pause for a question that Rui is probably going to ask.
Yeah. So no, I didn't want to really interrupt you. So we have a question from an anonymous attendee, which happens to be David Leclerc. So a dear friend and colleague of ours.
Yeah.
But so he's asking about the claims that Bacon Coulter has about the linearity of the APDs that allow you to, basically recalculate the matrix, the compensation matrix based on changes in gain settings, right? That you basically don't need to use the same gains for each single stain control. You can use different gains and it will recalculate everything so that it basically I'm missing the word, but it basically calculates the offset of those gains and so everything will match at the end, right?
Assuming perfect linearity of the, yes.
And I think
That's a phenomenal question, David, and it's something that's somewhat near and dear to my heart. So it's something that I personally don't feel comfortable with. I can't say that I've done extensive testing, right? So I have to say it's something that I don't feel comfortable with without doing extensive testing to be able to see if those compensation matrices that you obtain based on the assumption that everything is perfectly linear in response, assuming that that would be correct. Now, I have done some testing on linearity on the low end and the high end using 8 peak beads with gain changes, doing a gain tration. And I didn't have perfect linearity, which is what I want to see, but that's somewhat anecdotal, right? So I'm one person. I do know that in Cytometry Part A, there was a paper published. I know Rachel Sheridan and Tim Bushnell were on that paper, and it was about characterizing a CytoFLEX, and they do go into a bit more detail on that. That I can't speak with much knowledge to, but I know that there is some published references out there. For me and what I feel comfortable with, I only want people to run the compensation matrices those days at the same gains and apply those same gains and comp matrices to your experiment.
Yeah, I'll just add a little bit to that as well, Kathy. I'd probably agree with you. I think it's one of those things that, in theory, it should work, but the theoretical world and the real world are often different. And I think I'd need to do a lot of testing yourself to make sure that that was the case and you were convinced by it. I mean, it may be one of those things that in a few years' time, everybody will do it because it's perfect, but you do have to characterize your detector, whether it's a PMT or an APD, to show that it's perfectly linear.
Right.
Absolutely.
Yeah, I think the key thing is, like you guys said, is whether the instrument is performing as expected, right? Do we know it's performing as expected? Maybe not, right? So, especially outside of a core facility where the expertise is not as good, right? To understand if the instrument is performing well. And then what Kathy also was mentioning is that, at least in the context of a core facility where we're trying to teach best practices, it's also a bit hard to comfortably tell people, oh, for this instrument you can do that, for those other instruments you can't. And so we tend to just teach everyone as much as possible how to use best practices. And in all other systems, you need to make sure that you use the same gains or the same voltages, right? So I think that makes it a bit easier to make sure that everyone is doing the best for cytometry.
Yeah, absolutely. You could tell it's something all three of us are very invested in, as we both had, or all three of us had responses to that question. All right. So with that, I know the screen's a little bit messy. I probably should have taken that time to clean it up a little bit, but maybe that would make it a little bit easier for everybody. So I'm going to go ahead, because we have the time today, and just clean it up a little bit. This isn't going to apply a little bit later on, but just to try and clean up the screen a little bit, I'll try and move some of these. Okay. And I'll just cover some of the other ones. Okay. So the ones that are larger are the fluorescent detectors that we're interested in, and the other ones we're not going to worry about because they're not a part of our experiment today. All right. So I went over that we want to start at our slow flow rate, which is going to be 10 microliters per minute. And I change our events to display. And when we get there, when we need to optimize our forward and side scatter and our fluorescent gains, we're going to use that G hand. Okay. So now these are all of the dot plots and histograms that I'm interested in. And then what I'm going to do is I'm going to go ahead and hit run so that we can do a quick test to see what our signal looks like and make sure that our gains are all optimized. So I'm going to go ahead and hit run there. Should be on V2. And this is where there's a little drum going in the background, waiting to see our events. Something that you might want to be aware of, there's a pretty long sample line from the time of, or the length of the probe from the well to the flow cell is pretty long on the CytoFLEX. So sometimes you might see a tiny bit of a lag, and you can make sure that your boost is appropriate to see the signal. Okay, so as we're going along, we see here, I just adjusted my scatter gate, and we see two populations currently by forward and side. What I'm going to try and do is use my adjust gain hand to see if I could optimize that forward and side scatter a little bit. So I'm going to go ahead and I'm going to bring that up. I'm going to see what that looks like. Okay. All right, so here I see a population right here. I see a larger population. I want to see if we could pull out our lymphocyte, monocyte, and granulocyte population, because the nice thing about these TBNK synthetic controls, or a lot of the controls from Slingshot, is that they mimic the scatter profile of the cells that you're interested in. So for, in this case, TBNK, it's mimicking that TBNK cell profile. Okay, so right now what I'm seeing, if I look at my FITC, my PE, my APC, my C-Fluor 420 rather, and my APC, and my BB605, I'm seeing signal there. And what I want to do is I want to actually adjust this little scale right there to see if I can adjust my linear log display. Okay. Oh, I went a little too far. Apologies. Okay, let's see if I can see that. All right. Apologies, a little difficult on the screen. Oh. All right, adjust that a little bit. I'm just trying to optimize this display for these histograms. Okay. What I can do though, instead, what I'm going to do instead is I'm going to take this G hand and I'm going to see if I could optimize these signals a little bit. Right now they're between 10 to the third and 10 to the fourth. If I bring this up a hair, see where that signal falls, make sure everything's on scale, right? And I'm going to ask, yeah, go ahead. I'm just trying to optimize because I'm going to adjust the flow rate actually.
Yeah, I just want to make sure that in the fourth scatter, we're seeing the lymphocytes.
Exactly, yes.
Right, okay.
Yeah, let's bring that up a little bit more. And I think adjusting that flow rate actually helped a little bit because now we're seeing it in addition to maybe that slight modification of gain. We're able to now see that right here once we changed over to the medium flow rate. Okay, perfect. So now what I can actually do, based on what I know from my panel, what I'm looking to hone in on is my my T cell population and B cell population, which we expect to have a lower scatter. So right here, if I look, what I see is I see better profiles but on the histograms once I've honed in on this population right down here, which is where we expect to see everything by scatter, right? And I have my CD45 on scale for FITC. For PE, I have my CD4 on scale, and we see the 3 different populations. So everything's looking pretty good on scale. I might want to bump up the APC a little bit, and things are looking okay. So what I'm going to do for right now is I'm actually going to stop this run now that I know that all of my fluorescent signal is on scale. And I see Rui has popped up again. So I'll let him go ahead and interject.
This is a good question because it has to do with how you're increasing the gains and adjusting the gains. Somebody's asking if you can explain how you're able to move the peaks with no change in the x-axis values.
How I'm able to move the peaks with no change in the x-axis values.
They are changing, right?
Oh, so how was I able to make that change to what that visualization was? So if I'm understanding that for
when you're using the hand to basically increase the.
Oh, gotcha. So when I'm using that G hand, what's actually happening is if I open up this acquisition settings, all of these gains have been modified. So when I moved, for example, that AP, well, the red 660, right? That's our APC, which is what I expect to see for my CD19. When I took that G hand and I dragged that up a little bit, it actually modified the gain of my APC detector. So right now it's reading at 1007. And before that, it would have been slightly higher, or sorry, slightly lower. So because I dragged that peak up, it went ahead and it modified that gain to try and place that population based on where I visually wanted to see it. So that's one really nice aspect of this software and what we're able to do, because it is pretty powerful and we're able to modify those gains in that way. Okay. If that makes sense. And
Yes.
that's their question. Okay, great.
Does make sense. I just have another quick question just to make sure that everyone is also understanding. So Ezra was asking, why does CD4 have 3 peaks instead of 2?
That's an absolutely wonderful question. So these are actually like they're mimics, and what you see in PBMC populations quite often is that you have you know, a lymphocyte and a monocyte and a granulocyte population. Your monocytes do express CD4 at a moderate level, and I'm not an expert in these TruCytes, but I believe they are likely mimicking that. So you have a high level and a moderate level of CD4, which is typical to what you would see in PBMCs. And you can pull out your lymphocyte versus your monocyte population in that way. Great question. Okay. And that's my assumption based on these particles. So coming here, you can see if I had went ahead and I had included everything, it was much more difficult for me to be able to go ahead and see those populations, right? So if I wanted to have an understanding of whether or not all of my gains were appropriate and if I was able to resolve all these populations, having it set on a majority of my events by scatter would make it very difficult. But once I hone in on the expected population by scatter, we're now able to see that you know, our events are on scale by fluorescence, and we're able to see the different populations that we're expecting. Okay. The whole purpose of this initial sample run is just to make sure things are on scale and we get that best resolution. So we're not seeing many events right now, but from what we are seeing, we know that our gain setup is appropriate for population resolution. So I want to make sure I preserve enough of this sample that I could record it in our actual experiment that we're going to run after this compensation is done. So I'm not going to run anymore. What I am going to do, though, is I'm going to go back up to settings, the very top of my toolbar, and I'm going to go back to compensation setup. When I do that, what I can now do is I can change the location of this FITSI control. So this is our FITSI control. I can label it right here. And as opposed to B2, which is where I know my fully stained sample is, I'm going to change that to A2. And I'm going to set that as my sample well. Right? So now it went from B2 to A2. And what I'm going to do in just a little bit is I'm actually going to run and record my FITSI single color control and it's going to go to well A2, and we're going to overwrite the fully stained sample that we had just acquired. Okay. You'll also see in CytExpert, you have the capability of choosing the sample type. So you have the capability of choosing cell or bead. We are running the Slingshot compensation bead controls today. So I'm going to go ahead and select bead. We also have an unstained bead. It's not letting me run that. Oh, sorry, we don't have that one today, actually. So I'm going to skip that. We also have a CD4PE, and that's an A3. So I'm going to check off use on our PE detector. And I'm going to choose PE. And I'm going to go to bead and select A3 and set that as my sample well. Okay. So it's CD4PE, A3. I'm just checking myself as I'm going along. Then we have our APC. So that's red 660. That's also going to be bead. And that's going to be an A6. Anna and Chelsea are there at the CytoFLEX. So since they prepped it, if they see me making any mistakes, please feel free to go ahead and correct me. Then we have
Anna and Chelsea are doing that, Kathy.
Okay, perfect. I know that we're actually not going to run the unstained bead today. We're not going to do that because I'm going to bring that up in a little bit why we're not running it, if that's the correction.
Kathy, maybe you can answer this question. Can you trick the system and run the fully stained sample also if running tubes?
Yes, so that's actually something that I used to suggest, run your compensation in tube because it might be a little bit easier as opposed to doing everything in plate mode. But then logistically, you have to clean in tube mode and plate mode. So to try and make things a little bit easier, that's why I've swapped to this method most recently. But if you set everything up in tube mode, the first tube that you run, just have that be your fully stained experimental sample, and then overwrite it once you've gone ahead and optimized all of your gains with whatever the true tube was that's your first tube in your sample list. But very easy to do it in tube mode as well, and even easier than it would be here. Okay, so as we're going along, I said we don't have an unstained control today, our unstained bead. We're running these beads that actually do have an internal negative, so I'm going to skip it. The one thing that is unfortunate is it doesn't let me choose unstained bead after I've already went ahead and recorded something without an unstained bead control. So that was actually the reason why we're not going to run that unstained bead today. I actually physically cannot click it at this point anymore to add it on. And that is something that can happen. So theoretically, when you're initially doing your setup, you can choose unstained bead instead of, unstained bead as you're fully stained, and you have to choose another fluorescent detector. But that gets a little bit more complicated. So we're going to skip that for a little bit later if anyone has any questions on me going through that process. Okay, this is BV605 bead A5. Okay, so we have our FITSI, which is in A2. We have our PE, which is in A3. I'm going to skip forward. We have our C-Fluor 420, which is in A4. We've got our BV605, which is in A5, and then CD19, which is in APC. Correct my typo. And now I'm going to go ahead and I'm going to select OK. Now what it's going to do is it's going to reset everything based on how I moved everything. And it's going to regenerate all of those plots for each of those single color controls. Okay. So it reset everything for me because it didn't like the way I did it, and it modified it, which is perfectly fine. And what we now have the capability to check, and something that I like checking every time I do something like this, is checking to make sure our gains have transferred over and are consistent throughout all the samples. So if I go down and I click to my PE, I'm seeing that none of these gains have changed. And I usually just spot check to make sure it's carried on through the rest of the samples, which it should, okay? But it's never a bad idea just to check. So your eyes are pretty well trained to notice any minor differences. So we would have seen any integers that had changed as I was clicking throughout that, which I did not see. So then what we're going to do is I'm going to go ahead and we're going to, I'm clicking on initialize right here. Okay. Okay. Go back up to my FITSI single color, and then I'm going to go ahead and I'm going to select run. It's going to ask me if I want to overwrite this data, which I do because I don't want my fully stained multicolor sample as part of my compensation experiment. So I select run and overwrite, and we should see that data now in just a second. Okay, start. Okay, and there are our beads. Okay, beautiful. So I adjusted my scatter gate, and I see my FITSI population is showing up just like we expect, our positive and negative. I'm actually going to go ahead and select stop, because what I want to do is I want to do a quick check of all of my single colors to make sure everything is on scale there as well. You want to make sure that you optimize your single color, or sorry, optimize your experimental sample and make sure that the gains are optimized on that. But then I do a very quick check to make sure all of my single colors are also on scale, because if one is way off scale and we can't gate on it for compensation purposes, we would technically have to walk it back as far as the gain goes to make sure everything's on scale. Okay. So then I'm going to go ahead here. This is my PE. I'll bring that histogram up. I'm going to go ahead and select run there. And I'm just going to check a little bit of each and every one. Something I mentioned, I believe, in the last class that we did is the color of this circle that's next to each sample. So blue 525, that's our FITSI. That's showing us blue. That's because it was recorded in run mode, and I didn't actually hit record, and it didn't hit all of its criteria. PE looks good, nice and on scale, so I'm going to hit stop there. These compensation controls look very good. I'm going to go ahead and I'm going to click on our red 660. We're going to test our APC. So yeah, that's an important thing as we're going through and checking to make sure these single color controls are on scale. You want to make sure that if you select run, that you do go ahead and hit record at some point, because if you forget and walk away, what's going to wind up happening is it's going to keep going and going and going, because there is no stopping criteria for it to meet. And what can wind up happening is you can pick up air in your system, and that could be very frustrating to deal with. And we want to make sure that you don't have any error, because that could lead to instability in signal and loss of the ability to recover sample. But if that does happen, you could typically just prime the system a few times and make sure everything looks nice and stable, and then you're good to go. Okay, so we're looking good for all of these. Our seafloor looks very good. Our APC looked good. So the last one we're going to test is our BV605. And once I know that one looks good, what I'm going to do is I'm just going to record about 5,000 to 10,000 events for each one of these single color controls, because I just took up a little bit to test all of these gains. Okay. And what I can do if I want, I could go ahead, and as long as this BV605 is on scale, which it is, I can actually go ahead and start recording, because I know that these gain settings are appropriate for all. So I'm going to select record now. And, you know, what's really important to remember is if I did have to make a minor modification to my gain for this detector, say, what I'd have to do is go back into each one of those single color controls and make sure that the detector that I had adjusted the gain for is modified so the gain is the same on that detector for each and every single color control. And that's going to be incredibly critical. Okay. Beautiful. So what I just did as well is I modified this negative gate. So you have a negative population and we also have a positive population. So I modified those gates for both. This scatter, if I wanted to bring out a node or two and make sure it's around the whole population of compensation beads, what I could do after I make the modifications is I can right click and I can apply that gate change to all tubes so that I don't have to change it for each individual one. Okay, so that one now has the green icon next to it because it has recorded 10,000 events. Okay, so I'm going to go up to our seafloor single color control and I'm just going to go select run. It's going to ask if I want to overwrite. I'm going to select yes. And then once I see the signal come up and look as expected, I'm going to go ahead and select record. You'll also notice you have the capability to enable a time to record or volume to record. So if you only have a certain volume in your wells, you might want to say record X number of events or stop at this volume, taking into account the dead volume, which on CytoFLEX is 10 microliters for U and V bottom, and I believe it's 20 for the flat bottom plates. What you also need to recognize as well, though, is that there is a loss of volume through the boost function. Like I mentioned earlier, there's a long distance between the probe in the well and the actual flow cell itself. So in order to not sit there and wait for too long, in order to see your sample, there's a boost function that's carried out that allows for you to see your sample quicker. So you will lose, from my experience, about 30 microliters in that boost. So if you have 10 microliter dead volume, 30 microliter loss to the boost, you want to factor that into your sample prep and also into your recording. Okay, see a question in the Q&A.
Yeah, I was answering.
Okay,
I was
perfect.
planning to answer, but we can answer it live. No, Ezra was just asking the red and blue peaks that you show there together. It means that those lasers spill over together. I mean, it's not the lasers, but the emission of the populations are spilling over. And yes, it's showing where each of the population will fall in the other channels, right? So
exactly, and it's showing it kind of like an overlay, right? So if that's how you visualize some data in other software, like, you know, it's showing you the overlay of the populations and where that shows up. So as we'd expect on our APC single color control, we're seeing some spillover into a multitude of detectors. And that's why you're seeing your positive there and then your negative population as well. Where we don't expect to see any overlap, like say UV 405 right here, everything is overlaid on top of each other because we don't see that spillover. Absolutely.
And this is what Derek was showing in his slides.
Yeah.
You see that 3 peaks instead of 2, right? Just the negative and positive. That's that extra peak spilling over.
Yes, absolutely. Absolutely.
There's another question here. Oh, you're gonna ask, Derek?
No, I was just going to say that sometimes it's very useful, isn't it, to put all of those histograms up, because sometimes you get fluorescence or photons ending up in channels that you don't expect. So
exactly.
It's always useful to have all of those there.
Yes.
Right.
Absolutely.
Yeah, it's very important. I mean, we've been surprised over the years, right? When, you know, manufacturers introduced lasers and we didn't know that these fluors could emit in certain ranges, right? And be excited by different lasers, and suddenly
I think APC is a good example. It is reasonably
violet,
excited by the 561 laser as well.
Yeah. And the violet, right?
Yeah, the violet, and you see it in BV 650. Yeah, and that's something like people often go, "What?" And it's definitely something we see all the time for sure.
So one of the questions here, and this is an interesting question, what would you suggest we do if all our stain samples are expected to not express a protein and will therefore not have the antibody to check the gain initially?
Oh, so for your fully stained, you're expecting everything to be negative. If you have a biological positive control, that would be ideal. So if you can source some type of material, so if you have like knockouts, right, that you're testing, if you could test the the material that has not been knocked out, that would be perfect. If everything you expect to have negativity no matter what, you can honestly reference some of our older videos and maybe look at the characterization of your instrument to see where you're getting the best signal resolution typically for cells of a similar autofluorescence, and then use those voltages or gains on your instrument. That would be my best suggestion.
Mm-hmm. Yeah, the other question is from Kiara, which is related to the CytoFLEX. So she'd been told that CytoFLEX is kind of more permissive and copes very well with bubbles, even if you take the full volume of the well. Is this true? Have you heard of this?
If you're going to be running air for about 5 minutes or so and something's left in run, that's more why I was a little bit cautious and saying to make sure you carry out a couple of primes and just be mindful that you hit record. I think any instrument, if you just keep injecting air and the lasers are being directed at a flow cell that air is going through, it's not going to be great for that flow cell. But as far as getting rid of air, usually it doesn't have that much of an issue. You carry out a prime function and it's usually pretty clean. If you've been running air for a little bit longer, I'd suggest running a couple of primes and just checking the signal stability by time. So have a dot plot open and look at time on the x-axis and the fluorescent detectors on the y, and just make sure your signal is nice and clean. But that would be my suggestion, just to test that. Okay.
Yeah, thank you.
So, of course. So I'm just going through and I'm checking to make sure all of my gates are correct, which they look okay. One thing that sometimes happens is if your negative is on the lower end, you might have to modify the linear log coefficient to make sure that you're capturing the entirety of the negative population. So there's a little bit of a lag on my screen, so I apologize because that's why everything's all of a sudden going very, very narrow. But just be mindful that you want to check to make sure that your negative and positive roughly add up to 100%, because if you're cutting off part of that negative, it could be skewing that negative population MFI. Okay, so we're all good in our single color controls. So now what I can do is I can go up to settings and I can carry out a compensation calculation. Okay. When I click on compensation calculation, you're seeing your compensation matrices, and you're not just seeing it for the fluorochromes that you've selected. You're seeing it for all the detectors in the CytoFLEX. Okay. Then what I typically tend to do is I typically tend to save a copy of that compensation. And 0722. And that saves a compensation file, right? Then I'm going to go ahead and I'm going to select close. And I'm going to say file, close experiment. So again, this is different from a lot of other softwares out there where everything is integrated into one experiment. Whenever I make changes, modifications, I hit save. So I'm doing yes, but the compensation was carried out. And now we're ready to start our actual experiment. Okay. And we just have 2 samples. We have an unstained and a stained. So this will go relatively quickly. And we're going to reference back to a lot of the content that we reviewed in our last class. So I'm going to go ahead and I'm going to go to new experiment. And from here, I'll just call it OpenFlow compensation 100722. Pardon any of my miscapitalizations. And from here, we have a blank slate. So what we need to do is we need to go ahead and we need to generate a plate. We need to choose which detectors we're using. We need to set up our gating strategy. So everything at this point is almost from scratch. with the exception that we already have our gains optimized and we already have our compensation matrix done. So what I like to do now when I'm running in plate mode, if you see where I'm hovering, I'm currently hovering over the plate icon, is I click on that and that's where I tend to start. This icon right here where I'm clicking allows me to add a plate. If you have a plate template or a layout, you can go ahead and add from there, but we're going to go ahead and show you from scratch. And I'm going to make sure that it's our U bottom plate and select OK. And then my experimental wells are B1 and B2, and that's where I have my unstained and stained. And if I hover over this icon right here, you can see that setting is sample wells. If I wanted to incorporate cleaning or DI water wells into my run, I could also do that. I do suggest everyone takes a look and make sure that they test carryover if they're running sticky samples or if they have very low expressing populations and all of that. You want to make sure that you're not experiencing significant carryover. And if you are, explore backflush and mix options and incorporating some of those cleaning and water wells into your run. Okay, so now we have our setup for this experiment. Acquisition settings and these gains, I don't want to touch yet because we've already optimized that, remember? And I don't have a photographic memory, so I don't remember that off the top of my head. So I'm going to show you how we can import those in from our compensation. But first, what we're going to do is we're going to go to channel, and I'm going to select the markers that I'm using for this experiment. So it's CD45 FITSI. And then we have our CD4 PE. Oh, I didn't have that on the correct one. Sorry, I'm looking at a much smaller screen here. Thanks everyone for being patient with me. Okay. Uncheck everything else that we're not using. We have our CD19 APC. Okay. Our C-Fluor 420 is our CD3. Okay, our BV605, see Rui?
Yes, so while you do that, there's actually a nice question, a very important one, really, from Chris, where basically Chris has had experience with CytoFLEXs, the S and the LX. They only use that one. Or those, and then basically the question, I'll just read it, maybe it'll be easier. They started using the option to adjust comps to gains and reused older comps, but they were having issues. They were told the obvious, the comps need to be run at least on every instrument with the exact lot of antibodies, blah, 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 etc. So this was done with fresh comp runs on the same day, but still things weren't working well. Many parameters were over or under comped, especially with viability dyes. They use the CytoTrol cells for viability. And then the Beckman-Coulter rep said that the CytExpert, the software, tries to comp well, but they say they feel it fails just as well. So they gave up and started doing it manually, which is obviously not ideal and very subjective. So because of since my panels have multiple 12 color stains, I don't have time to go through all the MFI values, but I don't trust the comp the instrument software calculates. So, so essentially they're having a lot of issues with trying to comp with different gains, etc. And even running the fresh controls. I would just say a few things, Kathy, before you answer. But so the important thing is that even when we have everything set up, the instrument is well set up, is performing well, even if the reagents are, supposedly working well and performing well. For instance, if you're using beads versus cells, you may encounter some differences in the spectral properties of each fluorochrome. So the fluorochrome won't be exactly the same in the bead that it is in the cell or vice versa, right? So that's the first thing. The other thing is, if you're struggling with compensation with this software, you can always load this into another software, try that, and see if the problem is coming from the actual software, which I I doubt, but again, it depends on how it's calculating those differences in gains, if you are using different gains for the different controls. But if you're not using, then the calculation of compensation should be really straightforward. Every software should be able to do that without any issue. You may have problems, again, because the controls are not ideal. That's a different thing, but the calculation itself should be straightforward. Any software would do that. So I would suggest trying just loading that data in another software and see if the compensation is different. If it is different, then there's clearly a problem with that software. But if it's the same, then there's something else. It's either the instrument or the controls that are not ideal.
Yeah, and I'll just add on to that. Those are all great points, Rui. I've definitely seen some comp issues running experiments on the CytoFLEX. I redo the comp with the same exact controls in third-party software, and everything replicates the same way. So it's typically, like Rui was saying, those controls, right, they're not as bright, it's not matched, things like that. And I think it drives home the point. You know, you need to run on each instrument, and the best thing is to identify those appropriate controls and run each time.
Yes, and there's a question from Ezra, but about isotype controls, which is a bit outside of the topic here. So I'll just answer, Ezra, if you don't mind. I'll answer in writing, so we can go ahead and.
Especially because I've been chatting so much throughout this that we're running a little bit low on time, but I like more time in the instrument anyway, so it's great. So here I just set all of my channels, right? So I set my labels and I unchecked any detectors that I'm not using. And then from compensation matrix, all I have to do is go to import. Okay. And oh boy, here we go. Compensation is the one that I'm going to go to. Testing my eyesight. I'm becoming an old lady. The top one is the one I'm going to go with. That's compensation OpenFlow final 10.7.22. That's the one I just saved. So I'm going to go ahead and find that .comp file, and I'm going to select open. And when I do that, this is the window that comes up. And this is what we were just talking about. What we want to make sure that we do is make sure that we're importing our comp matrix and our gain. You don't want to run at different gain settings because you've already optimized based on your experimental samples. So we need to make sure that we're running on this specific subset, or sorry, on this specific choice. So we're importing the comp matrix and the gains that we've already optimized. And I'm going to select OK. And you can see here, this is our comp matrices. Okay. From there, I'm going to go to stopping rules. Right now, we don't have any gates. So if I wanted to make sure I'm recording 100,000 events and I added a 0 there, that would be all events because I currently don't have a gating strategy set up, but we could always go back and modify this. Yeah, we could also do volume to record or time to record. So if I say, I don't care about time, but I want to make sure I'm not recording more than 100 microliters, I can select OK there and select OK. And it's going to give me this warning that I don't have time to record, and that's fine. So I'm going to select yes, I want to continue. Okay. Here you can close out of this window. You can keep it open. I'm going to close it up to give us more space. You could always add a sample ID. So B1 is our unstained and B2 is our stained. And I'm going to go ahead and I'm going to start a very basic gating strategy. So I'm going to add a dot plot here. I added 2, I double clicked. Okay. I'm going to make this window, make that plot slightly bigger. I can't tell you how grateful I am that right now I have a mouse and not a trackpad that I'm working on. Make things a little bit easier. And I'm just going to start off with this forward and side scatter. I'm going to initialize, and then I'm going to set it to medium. And what we're going to do is we're going to run and record our our unstained control rather. And I'm going to set up our gating strategy based on that unstained control. So I selected B1. I'm going to select run. I'm going to make sure our events to display is optimized. So I'm going to have that set at 20,000. Okay. And I'm going to go ahead and choose my polygon gate option right here. And I'm going to go ahead. I'm going to wait till I see the population show up the way I'm expecting them to. Maybe I'll set that flow rate a little higher. Okay, and I'm starting to see my populations. If I wanted to, I could adjust that threshold. Remember, we talked about this in our previous class. I could adjust that based on visual inspection, right? So if I say, all right, anything like here or below is debris, you could do that. And now we're seeing our 3 populations a little bit clearer. I'm going to select this polygon icon. I'm going to go around my expected lymphocyte population. And now that I have that gate set up, what I'm going to do actually is I'm going to change my events to record to P1 because that's the scatter gate that I want. And for this, I'm just going to select 30,000. I'm not sure that we have that many, but we'll go ahead and see if we can get that for purposes of today. And then we're going to go ahead and record. I'm going to keep an eye on that event rate and make sure it's not going too low because that means I might be towards the end of my well. Okay, and then I'm going to continue on with my gating strategy. So I'm going to select another dot plot. Okay. And then here I'm going to make it show me only the gates from, or the events rather, from gate P1. And then I'm going to do side scatter area. Click on the y-axis, go down to height. That's showing up there. It's not showing up for me. Sorry about that. I'm going to go down to height. A little bit of a lag on my end. All right. I'm going to ask Anna or Chelsea, if you are there and you have the capability to choose side scatter height, I'll let you do that. If not, we can skip the singlet gating for today because it's not letting me do that. Thank you very much. I appreciate that. Okay.
Kathy, while you're doing that, let me just so Kara was asking one value in the comp matrix was yellow. I didn't see it because I was answering another question here. Do you go ahead anyway if it's yellow?
Yes, so we optimized our gains to pull out the population. So if you have a compensation value that's over 100%, I typically don't pay it too much mind. I'll typically try and optimize a panel. So if I notice that there are any issues, I'll revisit. But just because a compensation value is over 100% does not mean that's going to stop me from my run. Okay, so I wouldn't necessarily go back and redo everything.
No, I would agree with that. I think people get a little bit too hung up on the actual value, the percentage value of compensation. It's as long as you set the machine up correctly, and you've got the right controls, it is what it is, and we deal with it. You know, it is just a mathematical transformation of the data.
Yep, absolutely. Okay. So then this sample was all done in terms of recording. So then what I'm going to do, I'm going to continue on with this basic gating strategy. So next I'm going to choose CD45. And actually I'm going to go and I'm going to do CD45 and I'll do CD3. And I'm going to select where it says all events. And now I'm going to show P2 because that's my singlet gate. If I want to see my gating hierarchy, I can click on this little population hierarchy icon right there. Now, if I want my CD45 positive, CD3 positive, I would expect it to be somewhere around there, right? And then I'll do one more. And what we'll do is on this one, we'll look at CD4 and CD8, and then we'll look at our fully stained sample and see how it looks, okay? So this is going to be gated based on P3. We'll do CD4 on the X. And we'll do CD8 on the Y. One thing I also want to point out, if you look here on the sample list, you can see there's a grid associated with that. And that's because compensation has been applied from that plate setup, right? One thing before I record that stained sample is I adjusted the threshold for B1. So I want to make sure that's applied to the rest of my sample. So if I right click on that well, I can apply my acquisition settings to and make sure that that's applied to the remainder of my well. So now it's just the one. So that will change the threshold for that one. Okay. And then we're just going to go ahead and I'm going to change it back to medium. Going to select run. And then we're good to record and see our compensated data. Okay, there it is. Go ahead and select record. Yes. Oh, I had it select, I didn't change the stopping events though, so that's why it says 100,000 all events, which is perfectly fine. We're okay there. Okay. So that's recording. I might want to look at some more events, right? Maybe I'll look at 40,000. And as we're starting to record more and more events, what you see here is we see our CD4PE, right? And then what we also see, I'm going to just do a rectangular gate here, is we have your CD8 as well. Okay. So now this is showing us the compensated data. Again, Chelsea or Anna, if you're there, the linear log display is just getting a little bit dicey for me. So if you want to play around with those for the CD3, CD45, and CD4, CD8, feel free. But sometimes that helps me visualize the data a little bit better on the CytoFLEX. And it's just that linear log display around 0, right? So now we've officially gone through and we've, I think it might have run dry. So we're probably, I'm going to hit stop there. Know that you're adjusting some of those linear log displays. So sorry about that. I didn't want to run air through your system remotely from Cambridge or Leominster, which where I'm currently at. But in a nutshell, in the past hour, that's how we would go ahead and run a compensation experiment on the CytoFLEX using CytExpert software. Okay. So is there anything else we wanted to review today in CytExpert, or any questions that anyone had about this software specifically that we wanted to see? I know this isn't, these aren't actual cells that we're looking at, but these are the TBNK controls and they look pretty good, right? We can see these different populations pulling out. And it's something that I'm definitely interested in exploring for nice non-biological positive and negative controls for staining. So, something I'm pretty excited about, and also able to look at them to see how our compensation worked.
Well, this is everybody's last chance, of course, to ask any questions. But thanks for all the ones that have come through so far.
So there is a question. What are the two populations outside the positive population in CD45? I think I have. Yeah, exactly, those two.
I'm not sure if that's CD45 high, low, because I don't know enough about these particles to answer whether or not that's something that they've built into that, but that's something that you'll typically see. So I can't answer it with enough knowledge to say exactly what it is, because these are the TBNK controls. So I'll take that. I'll just take my response with a grain of salt that they might be trying to mimic high and low level CD45. I can't help myself. I'm just trying to.
I suppose if we put up a CD45 side scatter plot, that might help.
Oh, yes, there we go. That's absolutely it.
Yeah, that looks pretty much like it should do, really,
Yeah.
doesn't it? In peripheral blood.
Yep. Nice.
Yeah, so I guess
Right.
that's what they are, right?
Mouse control is tricky, isn't it? When you're
That's
many
the only
miles
thing
away
that's a
you
little.
are from.
Yes.
Exactly.
So
Here we go.
Chelsea was saying also that there is a bit of lag since we're doing this through Zoom, right? There's a bit of lag, so they were having also a bit of difficulties in adjusting the scaling. So
Yeah. But it looks pretty good here, right? Like I could optimize this a little bit more and visualize those populations a little bit better. But when we're looking at everything, it's probably actually slightly over comped potentially. So we'd have to go, like I could go back and we look at how I gated those single colors to optimize that a little bit, because we see a little bit of a tail over there. But the nice thing about optimizing that linear log display is you can look at the data and show the appropriate scaling to pull out those populations, but not too shabby. Any more questions?
Coming through there at the moment.
Maybe one more.
Thank you, David. Thank you, everyone.
Okay. Thanks, David. Yes, thanks.
Yeah,
everyone.
go ahead. Go ahead.
Shall I just share the screen, Kathy, just for the last few seconds?
So I will go ahead and I'll stop the screen sharing from the CytoFLEX. Or actually, Anna or Chelsea, can you stop your screen share on your end? And then Derek should be able to go ahead and screen share.
I can probably
Perfect.
just take over
Okay.
where
Awesome.
I have that power. Yeah, there we go. So hopefully we've answered all your questions. But if you this video will be, well, it's been recorded, so we will put this back onto the OpenFlow cytometry site on YouTube in the next few days, which you'll be able to see this all again, should you so desire. Obviously, we will try to run another one of these at some point in the near future. We don't actually have a date yet for that, so if you keep an eye on the usual channels for that in LinkedIn and on Twitter. Yes, to be determined. But if you do have any questions, obviously, that you think about later on, you can email us at the OpenFlow Cytometry Gmail address. You can put comments onto the videos on YouTube. And of course, you can subscribe to the YouTube channel as well. So you'll get the information when we upload that. And you can follow at least myself and Rui on Twitter. We're still waiting for Kathy to set up her Twitter account. But
One
she'll
day.
get there in the end, I'm sure. So thank you all for coming. We haven't gone over too badly tonight, which is good. So yeah, thanks for coming, and hopefully we'll see you all on a webinar in the near future. Thanks, everybody.
Thank you.
Thank you,
And
everyone.
I quickly want to add too, I just want to say thank you again so much to Anna and Chelsea, and also Fang over at MSKCC uploaded and got a lot of videos onto Bilibili as well. So she spent a lot of time getting this onto another platform for us. So for any of our colleagues in China, reach out to us and we'll provide that link as well. Thank you.
Bye, everyone.
Goodbye, everybody. Thank you very much.